AS Level Biology 9700, Paper 1, Variant 2, October November 2024, question number 15 to question number 28. So, first of all, we will start from question number 15, and question number 15 is, which statement about phospholipids in such cell surface membranes are correct? So they have given us the four statements. Fatty acid tails allow most ions to pass through the membrane. The fatty acid tails are, they are hydrophobic in nature. So, whereas the ions are hydrophilic, so they will not allow the ions to pass through the membrane. So, this statement is not correct. Hydrophobic tails point inward facing each other in this way. So, I think this statement is correct. All polar heads face the cytoplasm. We can say that in this case, we can say that many polar heads face the cytoplasm. It's more accurate to say that they face outwards. So this means that the both towards the cytoplasm inside the cell and towards the extracellular fluid outside the cell. So according to say this is all. No, all, not all. So this statement is not correct. The phospholipids help with the flexibility of the membrane. Yes, it's true. The fluidity of the fatty acid tails allow the membrane to be flexible, and this is essential for processes like cell signaling, endocytosis, and exocytosis. So according to these statements, we can say the statement number 2 and 4 is the correct, and it means the option B is the correct option. A, B, C are not correct options. Now we are moving towards the question number 16. Question number 16 is, in an experiment, pieces of onion epidermis are put into three different concentrations of sucrose solution, P, Q, and R. So the pieces of onion are left for an hour and then examined using the low power of light microscope. So each diagram shows one cell from the epidermis that was placed, and each of the sucrose concentration. So these are the three concentration P, Q, and concentration R. So what explains the appearance of cell in solution P? And here, first of all, I am moving towards the concentration P. You can see the vacuole is larger as it is present in a normal cell. So you can see it is larger. So we can see that it is turgid. Its shape shows that it is turgid. It means the water tension of the solution P was more than this water tension of the cell. So water entered into it, due to which it is turgid and it is increased in size. Whereas we are moving towards the R, as you can see, it is plasmolized. It is plasmolized, as you can see it is shrinked because it shows that the concentration or the water potential of the solution R was lesser than the concentration or the water potential of the cell sap. So water moved outside the cell. So it is plasmolized and or shrinked. Whereas E, it is a normal cell. It is not showing any sign of turbidity or plasma shrinkage. So we can say that it is a normal cell. So there was no entry of the solution into the cell, or there was no, you can say, the water or the cell sap was not left from the cell sap. outside the cell into the solution. So the first option is the concentration of solution equal to the concentration of solutes in the cell cell. This is not correct because the concentration of the solutes or the concentration of solution X. So the cytoplasm, this is about the concentration. There will be no, we are talking about that the water will always move in that case when the water potential is high from one side to the other. So it will move towards that side where the water potential is low. So the cytoplasm has the same concentration of sucrose at a solution. Again, the water potential of the cytoplasm is equal to the water potential of the, it is not, the water potential of the cytoplasm is not, the water potential of the cell sap is equal to the water potential of the solution. So this is the correct option. That is the water potential of the cell sap was equal. So it is of normal size. There is no movement of water outside the vacuole, and there is no movement of the water from the solution into the cell sap. So in this way, I would like to say the D is the correct option. All other options are not correct because they have talked about the concentration of solution and the concentration of cell sap and the concentration of cytoplasm. But the osmosis when the water potential is high. So in this case, the water potential of the in the solution was high, so it entered. Whereas in R, the water potential was high into the cell sap, due to which you can see the vacuole is very small in size, so it is shrinked. So water sap, so the cell sap has moved outside the cell. So in this way, I would like to say D is the correct option. Now we are moving towards the next question. Question number 17. And question number 17, you can see the electron micrograph shows rod-shaped bacteria. So rod-shaped bacteria, they have shown us the rod-shaped bacteria, and they have given us the length, which is 2 micrometer, and the diameter of the bacteria is 0.5 micrometer. So we have to take these and we have to calculate the surface area to volume ratio. The actual length of the bacterium is 2 micrometer and the diameter is 0.5. So assume that the bacterium is cylindrical shaped. What is the surface area to volume ratio of the bacterium? For this purpose, we have to calculate first of all the surface area and then volume, and then the ratio between surface area and volume. So first of all, we will, I'm going to write the formula. Surface area is equal to 2 pi r plus h. And whereas the now we are going to surface area is equal to 2 into pi 22 by 7 into radius. Radius diameter is 0.5. So we will divide the 0.5 with 2. So it will be equal to 0.25. So 0.25 and 0.25 plus height is 2. So after this calculation, I have got this answer 3.53. Wait a minute, my mobile, my laptop is touch. So sometimes I don't want to write anything, but my without any, sometime it is written. So you can see 3.53 is the surface area. Now I'm going to find out the volume. calculate the volume is equal to, volume is pi r square h. So pi 12, 20, r is 0.25 square, h is 2. So after calculation, I have got this 0.39. Now we have to find out the surface area to volume ratio. So it is I can write surface area to volume ratio. So in this way, I can write volume. Surface area is 3.53 is equal to 0.39. If we divide 3.53 by 0.39, the ratio is 9 is to 1. So the answer is D. So we have find out the answer after calculation. Without assuming, we have calculated the values, and as a result, we have got the answer D. A, B, C are not correct option. D is the correct option. After calculation, we have find out the results. Now we are moving towards the question. Next is 18. And here we can see, yes, chicken has 78 chromosomes in the nucleus of a body cell. So how many DNA molecules are there in a chicken body cell at the start of the prophase of mitosis? You know, the prophase consists of 4 phases: prophase, metaphase, anaphase, telophase, and mitosis is a cell division in which the chromosome number is maintained generation after generation. So, if this chicken has 78 chromosomes, so as a result of mitosis, when there will be the two cells formation of gametes, both of they will have the both of the daughter cells will have the 78 chromosomes. As you know, the mitosis takes place in the somatic cells. So in the somatic cells, chromosome number is maintained generation after generation. So when the mother cell is divided into two daughter cells. As a result of their division, there is two daughter cells will have the same number of chromosomes as the parent cells has 78. So in this way, I would like to say that what happened during this, how many DNA? Now we have to tell they in the start, in the prophase, before prophase it has 78, but you know, before prophase there is interphase, during which the cell made its preparation for division, for mitosis, for cell division. So during interphase the cell replicates and chromosome number is copied to, it is become doubled, which is 156. Because this is the possibility when each daughter cell will have the equal number of chromosomes as the parent cell has. So parent cell has 78. So the each daughter cell will have the same number of chromosomes. For this purpose, in the interphase, the when cell is making its preparation for mitosis, for cell division, so at that time the cell chromosomes are replicated and they are doubled, that is 78 to 156. So the answer is 156. That is It is T. Now we are moving towards the next question. Question number 19. So here you can see a photograph. Photomicrograph shows cell undergoing mitosis. They have shown us the different phases of mitosis. So which statement describes what will happen next in cell X? So here you can see this is the X, cell X, and the first statement is chromatin coils up tightly and the nuclear envelope breaks down. It has been done. Now they are condensed, they are present almost in the center. So chromosomes line up along the equator of the cell and attach to the spindle. That will be the next stage in which is called as the metaphase. And during that stage, they will they line up uh, along the equator in the center of the cell. In the center of the cell, so they will attach themselves with the spindle fibers. So this is can be the correct. Sister chromatids move towards the opposite poles. So this is the they are talking about anaphase in which the chromosomes are separated from centromere and they move towards the opposite poles. So this is not the correct statement. Spindle fibers break down and cell prepares for cytokinesis. This is the last stage, telophase. And after telophase, the cytokinesis takes place in which the cytoplasm divide. So this is the stage, you can say, the late prophase. That is after this stage, the chromosomes, they will line up along the equator of the cell and attach to the spindle. So B is the correct option in this case. We are moving towards the next question. Question number 20. And question number 20 is which process occur in bone marrow cells that are in a mitotic cell cycle? Here you can see the three statements they have given us. Phosphate group bind to ADP molecules to form ATP. So formation of ATP takes place when phosphate molecule will bind with the ADP adenosine diphosphate. There will be the formation of adenosine triphosphate, which is called as photophosphorylation, or we can say the phosphorylation in which the ATP formation takes place. And this ATP formation will be used up for the mitotic cell cycle. So I would like to say this is the correct statement. Bonds form between nucleotides in a DNA strands. This describes DNA replication, which is essential for mitosis. So before a cell can divide, it must duplicate its DNA so that each daughter cell receives a complete set of genetic information, as we have discussed in previous MCQ. So again, this is the correct option. Now the third one is hydrogen bonds formed between transfer RNA anticodons and messenger RNA codons. So hydrogen bonds. So I would like to say this is also correct. That is the hydrogen bond formation between transfer RNA. So bond formation will take place between and messenger RNA codons. So this is again the correct option. So according to this, A is correct. B, C, and D are not the correct statements. So we are moving towards the next question. The enzyme Telomerase. Telomerase prevents loss of telomers after many mitotic cell cycles. Which cells need to transcribe telomerase enzyme? So, which cells need to transcribe telomerase enzyme? Stem cells. So, what are stem cells? Stem cells, they they have ability to self-renew and differentiate into specialized cells. So they undergo frequent cell division to maintain their population and produce new cells. So telomerase is essential in stem cells to preserve telomere length and ensure their long-term viability. So the telomeres are the protective caps at the end of the chromosomes. There are chromosomes and telomeres are there. So they protect the DNA from loss because after every replication, the telomeres are lost instead of DNA. So telomeres are there. So stem cells, they need telomeres. So second type of cells are activated memory beta lymphocytes. Activated, the memory beta lymphocytes. They also need the telomeres. So enzyme telomerase. So the memory beta cells are part of the adaptive immune system. When they encounter an antigen, they rapidly uh, proliferate to produce antibodies. So you can say rapidly they require telomerase activity to maintain telomere length during the initial expansion of the memory cell population. They need telomeres because it should not be so lost. So they also need. Whereas there are helper T lymphocyte secreting cytokines. So here you can see these cells, T helper cells are, they also, you can say part of the adaptive immune system, but they do not need telomerase as much possible for them. So they are not needed by it. So according to this, I would like to say statement 1 and 2 are correct and 3 is not part of the answer. So B is the correct option in this case. Now we are moving towards the question number 22. And question number 22 is a polypeptide molecule contains the amino acid sequence glycine, leucine, lysine, and valine. So the table shows DNA triplets for these amino acids. You can see the glycine shows three nitrogen bases: cytosine, 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 leucine, guanine, adenine, adenine, lysine, thymine, 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 and valine, cytosine, adenine, adenine. So which transfer RNA anticodons are needed for the synthesis of this polypeptide? Transfer RNA. Okay, so they are DNA triplets. So we have to transfer RNA. The RNA single codon, and there is difference between the DNA and RNA in DNA. Guanine, cytosine, thymine, and adenine. These are four nitrogen bases. But in case of RNA, guanine, cytosine, adenine, and instead of thymine, there is uracil. So uh, it will be same and on uracil instead of it. So according to RNA, transfer RNA, the anticodons will be the option B because there will be only change in the uracil instead of thymine. All other will remain the same. Or Other types of nitrogen bases are same in both. Only there is change, that is thymine will be present in the DNA, and in RNA instead of thymine there will be uracil. So according to this, B is the correct option. Next question is 23, and you can see question number 23, they have shown us nucleotide molecules, a small strand of DNA, you can say. Which type of bond is labeled X? Here you can see the different types of uh, cells they have told us about these cells. Uh, glycosidic bond. Glycosidic bond is formed between glycosidic bond is formed between two carbohydrates by the process condensation reaction. Removal of one water molecule, the glycosidic bond is formed between them. So we can say that this is not the correct option. And hydrogen bond is formed. Um, you can say this is the dipole-dipole interaction between the molecules, as for example, between hydrogen and oxygen. So again, this is not a correct option. And peptide bond is formed between the amino acids to form a protein molecule. So again, this is not the peptide bond over here, but you can see this is a bond which is formed. You can say this is pentose sugar, pentose sugar, nitrogen base, and you can see this is the phosphate. So this is the bond which is formed between pentose sugar is the phosphodiester. So D is the correct option. So this is the phosphodiester bond which is formed between a phosphate molecule and pentose sugar. So this is phosphodiester bond which is formed in DNA. Now we are moving towards the next question number 24. And question number 24 is, which row correctly describes cytosine? Cytosine, cytosine is a nitrogen base, and this nitrogen base uh, is a pyrimidine. Nitrogen base cytosine is a pyrimidine. So pyrimidine type of. So number of hydrogen bonds with its complementary base are three. This is the cytosine with guanine. So it makes triple bond. So triple three, and we discuss its structure. So its structure will be a ring shaped structure, just like so. I would like to say it is a single. So this is cytosine. So single. So according to this, the question number 24 has the correct option. It's pyrimidine. This is fact. You know, cytosine and adenine. They are the pyrimidine. Now we are moving towards the next question. During the production of protein molecules, only one strand is the double helical used, which name is given to the DNA strand that is used to produce a new protein. And you can see that there is the formation of two strands, and one strand of DNA will act as the template strand, which is copied by messenger RNA. So it will be a template strand. And non-transcribed strand, the other strand which is not used for as a template strand, will be called as transcribed strand. So this is not the correct option. And leading strand is that strand which in which the you can say the copying is start from the, you can see the primer start from the 5' towards the, its direction is from 5' towards the 3'. Whereas the other strand, you can say the lagging strand, you can say it is on in the form of the fragments. So in this way, I would like to say leading strand, this is not lagging strand, only the template strand is there. The strand of the DNA, which is copied at the time of transcription. So here the C is the correct option. Some are the, the factual answers. They consist of facts. Now we are moving towards the next question, and you can see over here the diagram shows a longitudinal section of a flowing sieve tube with a companion cell. This is the sieve tube element, and this is the companion cell. What are the mitochondria? Where are the mitochondria located for the release of energy for transport? A is, you can see, lumen of the phloem. There will be no cytoplasm over there, so no mitochondria over there. A is not correct option. C is the membrane. So again, this is not the correct option. C, plate, not a correct option. So only cytoplasm of the, the companion cell have a proper nucleus. It has proper cytoplasm. So mitochondria exist in the cytoplasm. So B is the correct option. You know, there are no cell organelles in the sieve tube element or the phloem. So A is not the correct option. C again not correct option. So the mitochondria only be available in the cytoplasm of the companion cell, because the companion cell is a complete cell, is a living cell with nucleus. Now we are moving towards the question number 27. And again, this question number 27 is on the diagram shows a phloem sieve tube element and a companion cell that are involved in translocation of sucrose. And the phloem sieve tube element and companion cell, the phloem, and both of they are helpful in the translocation of the sucrose. Uh, which process correctly describes the translocation of sucrose through these cells? So we have to find out the correct answer. So the first option is the first option. Before going, I think detail, I think I would like to say something about these. That is the phloem is that plant tissue that transports sugar, sucrose, amino acids from source to sink. Source where the food is produced Source to sink. Source where the food is manufactured, and sink all other parts where the food is needed. Or sometimes you can say the stored. So the sieve tube elements, these are the flowing which are connected end to end in the form of long tubes, and they have sieve plates as in the last MCQ it was shown, and the companion cells which are associated with the sieve tube elements and perform various metabolic functions including loading and unloading sucrose. So the active transport, the movement of molecules against a concentration gradient by using energy, and diffusion, the movement of molecules from the region of higher concentration towards the region of lower concentration. So in this way, I would like to say these are the different processes involved in the translocation of sucrose. So first option, sucrose moves from Y to Z. Y to Z, you can say there is not by active transport, the movement is by mass flow. You can say or the mass flow and or bulk flow. So this is not the correct option. Protons move from Y to Z by active transport. So the protons movement, you can say it's coupled to sucrose 4. So I would like to say that this is the correct option. As you can say in Y, they are in lower concentration as comparison to C. So they will move from its lower concentration towards the higher concentration. So it is the uh, correct options. Protons move from Z to the protons, they will move from Z to Y by diffusion. So here I would like to say that uh, this is also incorrect. Protons are more likely to be involved in co-transport with sucrose cross cell membrane. So which is an active transport, not the diffusion. So already told they move Y to Z. They do not move from Y to Z to Y. So this is opposite of the correct statement. Protons move from Y to X by diffusion. They will not move by diffusion. They move by active transport. So again, this is not the correct option. Only B is the correct option in which the protons move from Y to Z by active transport outside the companion cells. So uh, this is the which type of molecules are transported into companion cells. Which type? Monomers and disaccharides. Monomers consist of single, as for example, you can say the amino acids, glucose or sucrose. Sucrose is a disaccharide. So in this way, this can be the correct. Monomers and polysaccharides. Polysaccharides are you can say the starch molecules. Starch molecules are formed when the glucose molecules are joined together to make polysaccharide molecules for storage purpose. So the starch is not translocated. So this is not the correct option. Polymers and disaccharides. Disaccharides, these are the carbohydrates. Polymers are not the carbohydrates. Food is manufactured in the form of carbohydrates, not in the form polymers, can be the any type of larger molecule. So again, this is not the correct option. Polymers and monosaccharide not. So only A is the correct option in this case. So I would like to say the A is monomers and disaccharides. These are the molecules which are co-transported into the companion cells. So it was all about from question number 15 to 28. Please like, share, and subscribe my channel. Thanks for watching.